So hello everyone, welcome to the CAC lecture. So today we have Yohan Yi. So Yohan joined the CoBra lab in 2022 as a postdoctoral fellow after completing his PhD within the Department of Medical Biophysics at the University of Toronto. Prior to that, he completed his undergraduate studies in McMaster University's Integrated Science program with a focus on physics and mathematics. Yohan's PhD work, which was carried out at the Hospital of Sick Children's Mouse Imaging Center, examined the neurobiological correlates of structural covariance networks. As part of his work, he has extensive experience working with multimodal imaging datasets on brain structure, gene expression, and connectivity. Within the CoBra lab, Yohan will work with mouse models of demyelination in order to examine the relationship between myelin concentration and functional connectivity. So thank you, Yohan, for being here today. And I'll let you begin.
Awesome. Get a round of applause before. Okay. So thank you for the wonderful introduction. I'm told I speak quietly, so if you can't hear me at the back, just let me know to speak up. As Liz mentioned, I'm a postdoc here at the Douglas, but what I'm going to talk about is my PhD work on structural covariance that was done at the institutions here at the bottom left. Just so we're on the same page, I'll give you a bit of a background on what structural covariance means. And by structural here, I'm talking about brain structure. So brain structure, brain structure in terms of volume and cortical thickness is important because it really encodes a lot of information on things like genetics, on the environment, and on states of disease. A cool example that I like to share about brain structure is in the hippocampus, where the size of the hippocampus tracks with the amount of spatial learning and the time spent spatial learning. So this was done in a study on London taxi drivers. A more salient example that's likely genetic in cause is agenesis of the corpus callosum. So in this case, you can actually see the lack of an entire brain structure in an individual MRI scan. But brain structure doesn't really tell you everything. We know that the brain works by connecting different regions, and these different regions function by sending information from one part to another. And this works at different scales. So at the microscopic scale, cells are connected to each other. In the cortex, they form cortical columns that we think are sort of fundamental computing units. And at the macroscopic scale, you can measure connectivity in terms of structure using modalities such as diffusion-weighted MRI and tractography, which we call DTI, or functional imaging modalities such as fMRI and MEG. It turns out that you can actually measure structural networks just using properties such as volume and cortical thickness by correlating their values across individuals. And so this is what we call structural covariance. So to show you how this works, I'm plotting the volume of one brain region against another. Each point here is an individual subject, and the strength of structural covariance between these regions is represented by the correlation. So in other words, individuals with a bigger volume for one region will likely have a bigger volume of another if structural covariance is high between these regions. A classic example comes from a human study by Jason Lurch and colleagues. So in the human brain, there's a part of the brain in the frontal lobe called Broca's area that's related to speech production, and another region that's called Wernicke's area in the superior temporal gyrus that's related to language understanding. If you look at which parts of the brain correlate in thickness across individuals to Broca's area, you find high correlation in Wernicke's area. So in other words, people with a thicker Broca's area tend to have a thicker Wernicke's area and vice versa. Now this kind of makes sense. These two regions are connected as part of the language network. And so you can imagine things like language fluency or language experience driving the thickness in both these areas at the same time. So in the words of this famous Canadian neuroscientist from the city, regions that fire together and wire together might also grow together. So a lot of people have actually looked at structural covariance in relation to wiring and firing. A study by Gao and Dong from 2012 looked at the association between structural covariance and structural connectivity as measured using diffusion tensor imaging. And they found that about 35% of regions that correlate strongly together were connected by a fiber trap. And then using multivariate decomposition methods, Segal and colleagues showed that structural networks, structural covariance networks, essentially associated very highly with functional components. So this has led to what I call the wire together, fire together, grow together hypothesis, where two regions, if they're connected, they basically, if one region activates, that results in activation in another region. We know from animal studies that consistent activation of a region can result in things like synaptic plasticity, which in turn can result in a volume changes detected through MRI. And so these two regions are connected and these processes occur in a coordinated manner. You'll measure this as a covariance. That isn't the full story though. We also have another similar, but not necessarily the same hypothesis, which essentially says that regions might just grow together. You don't need connectivity. What you can have is just coordinated development that's guided by intrinsic genetic cues being expressed. And so evidence for this comes from a recent study by Sophie Bach, in which they decomposed structural covariance into genetic and environmental components. And they did this using a set of twins. You need a twin study to do this. And they found that the genetic correlations were much stronger than the environmental correlations. And if you look at simply patterns of coordinated development, so this is called maturational coupling, you find that structural covariance networks are very, very similar to that. So in other words, if you have two regions, they share the same patterns of genes, they express the same genes, they undergo the same developmental processes, they'll likely be coupled together in their growth patterns. And at a single time, if you measure their volumes, you'll measure their correlated volumes as a covariance. Yep, Bashar has a question.
So you're assuming they're not connected.
They can be. So both these hypotheses overlap in some sense. So the question for those on Zoom was that we assume they're not connected. In this second coordinated development hypothesis, the regions can be connected. If a pair of regions, they share similar genes, they might also still form connections. So just to clarify, these aren't necessarily completely independent. There is some overlap between these hypotheses. In the literature, this idea of using structure to form networks has been very appealing. And as you can see in the top plot, the number of studies plotted over time has really shot up over the past decade. But what we really don't know is what's underlying the formation of these structural covariance networks. So back in 2006, in that study that I cited earlier, Jason mentioned that the biological causes underlying correlations between cortical thickness, it's not really well defined. A few years after that, in a very influential and highly cited review on structural covariance, Aaron Alexander Block says that their questions still remain as to the biological interpretation of structural covariance. More recently, the underlying neurobiological correlates of structural covariance is still very ambiguous. And most recently, again, the exact mechanisms that underlie structural covariance networks still remain uncertain. So a lot of people have used these networks to probe development, to look at differences in disease, but we don't really understand why brain regions couple together in their volumes. I've highlighted this last one here, which really speaks to the wire together, fire together, grow together hypothesis. And this is still one of the things we think is potentially driving structural covariance. So we wanted to ask a question, which is wiring really required for structural covariance? We decided to address this in the mouse because in the mouse, we have a lot of genetic and environmental control. We can use inbred mice, which essentially have the same genetics. We can control their environment. We can generate large sample sizes, and using high resolution MRI, we can really look at very fine brain structure. Mice have a short developmental period, so you can really track development and coordinated development over time, right from birth to adulthood. I won't mention it in this talk, but we've done a lot of studies on that. And most importantly, in the mouse, we have data sets on gene expression and connectivity, which we can then compare our structural covariance results to. So that brings up a question. Can we detect structural covariance in the mouse brain? The way we would do this is, well, first we need to get volumetric information. And to do that, we need to scan the mice. All the mice that I'm going to talk about in the first two studies that I'll mention were scanned in a Varian 7 Tesla magnet at the Mouse Imaging Center. They were all scanned ex vivo, so 16 brains at a time. We put them in this basket of 16 radio frequency coils. The brains are left in the skull, and we use a T2-weighted fast spin echo sequence. We do this overnight. It takes about 13 hours or so to run the scan, and you get back images at around 50 microns in resolution. So after the brains come out of the scanner, after the images are reconstructed, if you align them linearly, so we're just rotating them, sort of putting them in the same space to compare, they look like this. We're going to use a process called deformation-based morphometry to compute local volumes at every point in the brain. And the process works sort of like this. What we need to do is we need to align the brains non-linearly to look like each other, essentially. And to show you this, I'm going to overlay a set of grid lines on each of the brains. There are 4 brains here. I'm showing you 3 different brains on the left and 1 additional one on the right that's zoomed in, so you can sort of see this warping process. And when you warp them to look like each other, you can see the grid lines sort of either come together or expand. By computing this warping process and overlaying a set of regions or an atlas on top, what you can do is then compute volumes. So I'm overlaying a mask for the motor cortex at the top and the hippocampus here. And essentially, by summing up what we call the Jacobian determinants, and I'll mention this in a second, but by summing them up under these and weighting them by the voxel volume, you can compute the overall structure volumes. And then finally, if you do this for multiple mice and plot one volume against another, you can get back a value, a measure of structural covariance between these pairs of regions. You can also do this at the voxel level. And so to understand how volume is encoded at the voxel level, I'm zooming into 2 parts of this mouse brain. One that's labeled in white, where sort of the grid lines come together. And so what this means is that for this mouse in particular, this part of the brain highlighted here or in white had to shrink to meet the average. And so what it means is this part of the brain had to start off bigger than the average. And so this shrinking encodes volume information. Similarly, the lateral part of the cortex in this brain had to expand to meet the average. And so it means it started off smaller. So quantitatively, at every point in the brain, we encode this volume as something called the Jacobian determinant. And then what you can do is pick one of these points in the brain as a seed, and the other one, we call it the target, and we can correlate the to very local points. If you vary the target across the brain and compute the correlation for every point in the brain with respect to the seed, you can generate what I call a Voxel-wise structural covariance map that essentially says how similar do these other parts of the brain correlate in volume to the seed. So for a seed in the hypothalamus, a structural covariance map looks like this. Essentially, you have high correlations in the hippocampus, you have high correlations in the cerebellum, and you have high correlations in the hindbrain. One thing you'll notice is that structural covariance tends to be very bilateral. So if you have high structural covariance on one side of the brain, you'll typically have high structural covariance in the other. So this answers the second question, which is, can we detect structural covariance in the mouse brain? We do see these fine patterns that reflect what we already know from human studies, that these patterns are bilateral and they kind of make sense when you look at which regions correlate together. So this then led us to 3 different studies that I'm going to talk about. In the first, we looked at the statistical relationship between structural covariance networks constructed in the mouse, and we compared that to neuronal connectivity assessed using viral tracers and to maps of gene expression similarly. In the second study I'll talk about, we used mice in which there's disconnectivity between regions, and we assessed whether structural covariance changes in that. And if I have time, we'll talk about a third model in which there's basically miswiring. Axons don't form the way they should, and we looked at structural covariance in that. So in the first study, we asked, what do structural covariance networks in the mouse brain overall look like, and how similar are they to neuronal connectivity? The data, it's all actually described in this 2018 study that we published in a table. But basically what you need to know is that all the mice were from the same strain, all were a single sex, they were all male, and all were adult mice. We had 153 subjects in total. We used an atlas of 318 structures, and we computed structural covariance between every pair of these structures. So what you're looking at are a structural covariance matrix. Structures in the right hemisphere are shown on the right and are labeled by a color scheme that corresponds to the slices shown on the right. And correlations between these structures and other structures in the same hemisphere are represented by a color. So red is, I should have put a color bar here, I'm sorry I don't have that, but red is high positive structural covariance, blue is negative structural covariance. And similarly for targets in the contralateral hemisphere, you have structural covariance shown on the right. Essentially what you see are large modules show up with structural covariance being high between cortical regions and other cortical regions. That's in this module right here. And in general, negative correlations between cortical regions and cerebellar regions. So, yeah.
Do you, when you account for things, what is this? I was just wondering what this matrix looks like. We've done for things like distance, and I don't know if you know that we done, or structural volume itself.
So the question was, what would this matrix look like if we account for distance and structure volume? Yeah. So we haven't explicitly regressed out distance from this matrix, if that's what you mean. Sure. But what we've done is we've compared maps of distance to structural covariance, and we've asked how much variation in covariance can be explained. And I'll talk about that in a second. But first, before I talk about distance, the topic that we really wanted to study was how does connectivity relate to structural covariance? And so to get connectivity data, we went to the Allen Institute's mouse connectivity experiment dataset. As a background on this data, the Allen Institute injected fluorescent adeno-associated viral tracers into a whole bunch of different mouse brain regions. The tracers, they travel anterograde, but they don't cross synapses. So they're really a measure of anterograde connectivity from the cell body, and they label sort of individual neurons. The data, so the injected mice were imaged using serial 2-photon microscopy, which we then aligned back onto our MR images. We selected only a subset of all the experiments that the Allen Institute puts out. We noticed that there was a relationship between the volume of the injection that they used and the distance that the tracer travels. And we really wanted to not deal with cases where too little injectate was injected into the mouse brain and the tracer just didn't travel far enough because of that. So we only considered experiments in which the injection volume was above a certain threshold. And in total, that resulted in 108 different injection experiments. So for each of these experiments, you can imagine there's an associated tracer image that tells you where a tracer would go if you injected in each of these different sites. Just to clarify, the data they consist of injections in the right hemisphere. So if you look at structural connectivity constructed from the injection data, you'll find that it's much more sparser. Just with a visual glance, you can see that overall, there's some structure to this that matches structural covariance. The large modules are sort of preserved, but overall, if you look at the patterns of covariance, what you find is that regions that are connected together, tend to covary, but there are many regions that are not connected together that also do covary across the brain. We also had gene expression data on hand, so we then decided to ask, well, why do these other regions still covary? Could gene expression explain that? So as a bit of the background on the gene expression data, again, the data is from the Allen Institute. They used in situ hybridization and profiled the expression of 4,300 genes that are expressed in the brain in their coronal data set. And essentially in this data set, they slice the brain in coronal slices, you get full brain coverage, and you get intensity values that tell you where is this gene expressed. What you can then do is, using this gene expression information, you can look at gene expression profiles and ask which pairs of brain regions have similar gene expression profiles. So we're constructing basically networks of transcriptomic similarity. And when you do that, you see that the networks look far more similar to your structural covariance networks. So again, if what these values represent are correlations, and if there's a high correlation between a pair of brain regions, what that essentially tells you is that the gene expression profile for one region is predictive of the gene expression profile of the other region. We wanted to quantify this overlap between connectivity, gene expression, and, as Mallar asked before, distance. And we did this in a Voxel-wise manner. So the data is quite heavy here. So I'm going to run you through what each of these data sets mean. So essentially, we have 108 different injection sites. And for each of these different injection experiments, we have structural covariance information. We have information on where a tracer would project if it was injected there. We have information on transcriptomic similarity, and that's shown here on the right. So going from the top to the bottom, the first two rows show you the background anatomy as coronal slices from the front of the brain to the back of the brain. And the seed region, one of these 108 seed regions, is shown here in red. The top row shows you anatomy using 2-photon the autofluorescence signal. And the second row shows you the MRI template that we used and we aligned to the Allen Institute data. The third row shows you structural covariance to the seed region. The fourth row shows you transcriptomic similarity to the seed region. So essentially, which parts of the brain have similar gene expression profiles to the seed. The fifth row shows you the tracer map for when a tracer was injected in that seed region. And the sixth row is a computationally estimated version of a tracer map if the tracer could cross a synapse. And finally, the last row here shows you the Euclidean distance to the seed.
The second or last row is one hop.
One hop, yeah, that's correct. Okay.
Did the Allen provide that computational distance?
No, we generated that. The way we did that was we looked at the overlap between the tracer projections and other injection sites, and then simply took the maximum merge of that. So the question that we had was, how much variation with the seed in the in the hypothalamus, we find that if we look at distance alone, you can explain about 20% of the variation in structural covariance. And I've denoted that as a little bar here on the right, around 20%. If you look at transcriptomic similarity, it looks similar, but if you could do the math, you find that about almost 0% of variation can be explained in structural covariance. Similarly, you can use your tracer image and compute variation explained. You find about 5% of variation is explained there. If your tracer can cross the synapse, it turns out that there's less variation in structural covariance that can be explained. So we did this for more than just one hop. We asked if the tracer could cross the synapse. Well, if it could cross two synapses, does that make things better? The answer is it doesn't. Very little variation is explained in that case. And so then we started combining predictors. So just to clarify, we're using a linear model here, and now we're adding multiple predictors. Together, transcriptomic similarity and distance explain around 30 to 35% of the variation. Similarly, you can do this for connectivity and distance with multiple hops across synapses, transcriptomic similarity and connectivity, and transcriptomic similarity and connectivity across multiple hops. And so we're really just computing multiple different models and examining how much variation can be explained in structural covariance. And finally, you can add all predictors in. So transcriptomic similarity, connectivity, and distance, and again, connectivity across this multiple synapses. And in total, for this experiment, we find that up to a maximum of, or almost 40% of variation can be explained. Now, this is just one experiment. We have 108 in total. And what you can do is represent that as a distribution of variation explained. We do that and we find that up to a maximum of around 40%, so that's the median in this distribution, can be explained by the predictors that we have. All of these are significant. The green bars are sort of the upper bound on the median that you can get by using a permutation test. But the important thing to take away from this plot is that not all the variation in structural covariance can be explained by connectivity. And gene expression in general tends to do roughly about the same as connectivity in explaining structural covariance. We looked at the variation in spatial patterns of structural covariance in the ipsilateral hemisphere initially. We also did that in the contralateral hemisphere, and we find that the values are about the same. We also divided the brain into different parts, and we clustered the data to see whether structural covariance or the variation that can be explained in structural covariance differs for the locations of the seeds. And in general, within the cortex, gene expression tends to explain structural covariance far better than connectivity. But in the subcortex, if your seed is in the subcortex, connectivity images look much more similar to structural covariance. So in summary, we find statistically significant relationships between connectivity and structural covariance, but it only explains a limited amount of variation. And we find that between connected regions, you often have high structural covariance, but there are many regions that are not connected, but you still have high structural covariance. So another way to put that is structural covariance is sensitive, but it's not specific to connectivity. So, many questions? Sure, yeah.
So did you use for the experiments only the exhibitory cell types, or did you use different cell types?
Yeah, so that's a good question. So the question was, did you use only excitatory cell types or all cell types? In this case, we used only the wild type injection. So essentially, the Allen Institute injected into, so let me take this a step back. So the injection experiments that the Allen Institute did were in both wild type mice and also certain Cree lines in which they essentially selected for certain cell types. we only subsetted the data to include the wild type experiment. So it's all the cell types that are captured when you inject in that region. Okay.
For the clustering, was there any weird outliers in that in the clustering that you gave the general idea, but was there anything interesting in terms of ones that popped out?
We didn't examine that too much in detail. So the question was, are there any outliers in the clustering? The input to the clustering was just the variation explained by distance, gene expression similarity, and your monosynaptic connectivity. So it's just 3 data points and 108 different observations. And we clustered those. We got 4 clusters. We found that this was the best solution. And you can kind of look at the distribution, the spatial distribution of the seed regions. So that's kind of shown in these renderings on the right. And if there were any outliers, I imagine that you'd find that within these images here. So to clarify, there was no spatial information on the seed that went into the clustering, but the seed regions do cluster within space together. What?
When you say like up to about 40% of variance explained by the inclusion of all those parameters in the model, can you use a sense of the order of magnitude of that? I know you said that we have that sensitivity, but that's not getting specificity.
Yeah.
But maybe like 40% of any norm measured.
Yeah, I think it depends on the context. So the question was 40% of variation explained seems quite high, particularly for neuroimaging, and that is true. To me, the context is how does it compare to other studies? And we typically see that when you compare structural covariance to other measures such as structural connectivity, so going back to that study by Gao Lan Gong et al., you get similar values in overlap, depending on your measure of overlap. So in their case, they used a binary overlap, and they found that around 35% of all highly covariant regions had a fiber tract connecting them. And so we're sort of in that same regime. Okay, so moving on to the second study, we found that there was this association between structural connectivity and structural covariance. It wasn't very high, but as Mallar mentioned, like these values are still reasonable for neuroimaging studies. And so we asked the question, is structural connectivity even required for structural covariance? For this, we went to mutant mouse models of disconnectivity. Some of the most famous models of disconnectivity involve agenesis of the corpus callosum. And the classic model for that is the BTBR mouse. So what you see here are 3 different models of 3 different BTBR models that we obtained from collaborators. On the right, the top row shows you the controls associated with that experiment. And what you see is a normal crossing corpus callosum. But in the mutant mice, the BTBR mice, the corpus callosum is notably absent. So there's no connectivity between the 2 hemispheres that pass through the corpus callosum, if it was present. So we had 3 different models of these, and then we also had an intersectin-1 knockout mouse model. Intersectin-1 is a gene that is required for fiber tracts to cross the midline. And when intersectin-1 is knocked out, you essentially have a lot of fiber tracts not crossing the midline. And so you get not only agenesis of the corpus callosum, but you also have a thinning or a lack of an anterior commissure. So the question that we asked was, between regions that would normally be connected by the corpus callosum, does structural covariance change in these models in which the corpus callosum is missing? To get to that first part of the question, which is the sort of regions that are normally connected by the corpus callosum, we use the Allen Institute viral tracing data again to find which regions are normally connected. So we searched through around, I think, 2,000 data sets or so, and we narrowed it down to 254 viral tracing experiments in which one side of the brain was injected and the tracer crossed to the other side. We had a few other criteria. For example, we wanted the tracer to be long enough to project far enough to avoid any distance effects. And we also needed the tracer to project far enough from the midline so that we didn't get any artifacts from image registration because we're dealing with mice without a corpus callosum, so you don't have that homology in that part of the brain. The image at the bottom shows you our mask and our labels corresponding to the corpus callosum, shown here in red. We additionally, from these 254 experiments that we identified connected different parts of the brain through the corpus callosum, we narrowed that down to 158 experiments in which the tracer not only went to the target on the other side of the brain, but also to a target that was ipsilateral in the ipsilateral hemisphere across the contralateral target. The reason we did this was to separate the effects of strain from the effects of connectivity. So the idea is that we'll compare structural covariance between our seed and our contralateral target to our seed and the ipsilateral target. Because we're dealing with BTBR mice, which are basically, they differ in genetics slightly to our controls. And so any difference that's genetically related should show up within this comparison. The examples for the different Allen Institute tracer injection experiments that we identified, so 4 are shown here, 4 different rows. The tracer, for example, for the top row, it shows you an injection in the claustrum, I believe, and you can see that it travels through slices that you do not see, but it sort of innervates 2 parts of the brain that are directly across each other. And there are 158 such experiments in total in which you can identify targets that are across each other, in which the tracer crosses through the corpus callosum. Before we compared structural covariance between these pairs of regions, we first confirmed again that in this data set, structural covariance is associated with connectivity, because that's something we wanted to verify. So as an example, for a tracer injection in the visual cortex here, you can see that the tracer, it crosses over through the corpus callosum. It's kind of hard to see here, but you can see that the structural covariance map shown on the left for a seed in where the tracer was injected, it looks quite similar. And we quantify this overlap between structural covariance and the tracer injections by binarizing both images and then computing the sensitivity. So here I'm binarizing the tracer signal at one value and binarizing the structural covariance signal at another value. And the sensitivity is essentially the proportion of bright green points or bright green voxels divided by the total number of green voxels. We can do this for every single experiment, and you get a distribution of sensitivity values. The sensitivity values for the experiment I've shown here on the left is outlined or is given by the red bar, and the median for this distribution is given by the black bar. We did a permutation test again to make sure that this median was significant. The permuted distribution of medians is given by the dotted line. So the story here is that in interhemispheric structural connectivity, structural covariance is associated with that. We also, because we're dealing with fewer mice and a different data set, we also wanted to make sure that structural covariance was replicable. We could compare it to our original framework where we computed structural covariance in a larger population of subjects. And similarly, we found that. So essentially in plotting structural covariance in control mice from the BTBR studies on one axis, each point here is a correlation. And I'm plotting that against the structural covariance value obtained from a much larger data set. And we're doing this over the different experiments that have identified. And here again, what you see is that structural covariance is replicable. So that brings us to the question that we want to address, which is how does structural covariance differ when connectivity is absent? So the plots I'm going to show you are a bit involved, so I'll run you through the hypothesis and how to interpret this. So the hypothesis is that if the corpus callosum is present, your seed region is connected to both the target region in the ipsilateral side and the target region in the contralateral side. If you look at if connectivity drives structural covariance, you'd expect that because these two regions are connected, the seed and the ipsilateral side, you'd expect to find a reasonable, moderate to high correlation. So this is just simulated data. And I'm plotting that as a point here on the right. In the case of agenesis of the corpus callosum, if you just look at the ipsilateral side, again, connectivity is present here. So you'd still expect there to be high correlation on the ipsilateral side in these acalossal mice. In the controls, if you look at the seed and compare it to the target on the contralateral side, connectivity is present. So you'd expect there to be high correlations. And again, you get a value that's reasonably high on the right. But in the BTBR mice that don't have a corpus callosum, if you look at the seed and compare it to the target, you would expect there to be very little to no structural covariance. So that there'd be almost no correlation here. And this value should be close to 0. And so the hypothesis here is that on the ipsilateral side, you should see no differences when you compare structural covariance between controls and the BTBR mice. But on the contralateral side, there should be a difference. This is just one experiment corresponding to one injection in a single seed region. You can do this over multiple different seed regions. If you do this for 4, I've sort of drawn the schematic here. And what you essentially would predict is that if structural covariance is driven by connectivity, you'd predict that there'd be a difference in structural covariance in the distributions only on the right. With real data, what we see is there's no such difference. What we did was first we initially pooled the controls from the 3 different BTBR experiments and compared that to the acalossal mice, again, all pooled together. We pooled them just to come up with a larger sample size, since that's an important factor in computing structural covariance. And as I mentioned, we don't see something that matches our hypothesis. In one of the BTBR data sets, we actually had BTBR mice that were genetically engineered to have a corpus callosum. So we did a comparison between only the BTBR mice that had a corpus callosum, and we compared that to BTBR mice that normally don't have a corpus callosum from that same experiment. So in this case, the genetics are identical. We're only looking at the effect of connectivity. And again, we don't see something that matches our hypothesis. The sample sizes are a bit lower, but here we're trading low sample sizes for a large number of comparisons across different viral tracing experiments. And finally, in the intersectin case, if you compare the intersectin mice, the controls against the intersectin 1 knockouts, we do see some change, some results that match our hypothesis, but there's also a slight decrease in the ipsilateral side that might point to ineffective genetic differences. So we did some basic tests, and we found that in general, we didn't find, but we also wanted to take a more principled statistical approach modeling structural covariance. So in other words, we want to model target volumes given seed region volumes across all these experiments. So this goes back to the 2006 famous paper by Jason Lurch, in which he established a modeling framework, and we essentially extended that to a linear mixed effects model because we're dealing with multiple viral tracing experiments. I've put the model here in the R programming language formula notation. It's a bit involved, but the idea that I want you to take away from this is we're essentially modeling the volume of the target as a function of the seed region volume and intercept, strain-related terms, hemisphere-related terms, and terms that relate to whether the target is connected to the seed or not. And there's also a random effects component that's shown at the top that has to do with the different viral tracing experiments that we're dealing with. So we trained the data on the BTBR data set in which we had both normal black C57 black 6 controls, but also BTBR mice that had a corpus callosum and that did not. And the idea was that this allows us to separate the effect of strain from agenesis of the corpus callosum. And so then using that, we can infer parameters for each of our terms related to structural covariance and related to the effect of agenesis of the corpus callosum interacting with covariance. The other two data sets allow us to test our model and assess the prediction accuracy and how important the predictors are. So we can take out predictors and see how well does the model do. So going to the first part on inferring parameters. Before I do that, actually, this is the structure of the data that goes in. If you're interested, we have around 8,308 observations corresponding to the number of mice multiplied by the number of different viral tracing experiments. So that's a substantial number of observations. Going to the parameter inference, we find that in general, we have a statistically significant effect of seed in predicting target volume. So that's your structural covariance effect. That makes sense. We would expect that. We find a significant effect of hemisphere interacting with seed. So that's essentially saying that predicting or the effect of hemisphere is important. Another intuitive way to understand this is that in the opposite hemisphere, structural covariance typically slightly decreases, and you're just measuring this effect here. There's also an interaction of strain that you can see as quite significant. But the term that we're really interested in, which is the effect of the lack of connectivity on structural covariance, we find that that is not significant. When you look at how well this model does on the test data sets, we find that the model actually does really well. For 3 different structures that span in overall size, we find that across the 2 different data sets, the structure volumes are predicted very well using our model. But when you start taking out terms from the model that I mentioned, this prediction goes down. So if you take out terms related to structural covariance, it turns out that you just can't really predict the overall target volume here very well. But if you take out the agenesis of the corpus callosum term, that's shown in the distribution on the right, that doesn't really change the variation explained in structure volumes. So that's essentially saying that structural covariance is present, but the effect of the lack of connectivity, it doesn't really play much of a role in predicting target volumes or in driving structural covariance. So in summary, we found that in interhemispheric structural connectivity, structural covariance, it's associated with that, but when you have the lack of connectivity, you still find structural covariance. And we're not quite sure why. One potential hypothesis is that there's potential compensation through other tracts. So maybe, brain regions, they don't wire together, but they still fire together because firing is preserved through things like the anterior commissure. That's something that needs to be tested. But just to go back to this idea of connectivity driving covariance, we did another study on a third model of, I should say, maybe like aberrant connectivity. Do I have enough time to describe this? A few more minutes? Yeah. So in this model, we use DCC haploinsufficient mice. So DCC, it stands for deleted in colorectal cancer. It's a gene that codes for a receptor for the axon guidance cue netrin-1. And it's involved in patterning the mesocorticolimbic dopamine pathways that develop in adolescence. So the idea is that in the ventral tegmental area, you have dopaminergic axons that develop and that go to both the nucleus accumbens and another set that passes through the nucleus accumbens and synapses into the medial prefrontal cortex. This switch between the nucleus accumbens and the medial prefrontal cortex is driven by the expression of DCC. So if you delete or if you have less DCC, you essentially have fewer neurons going and synapsing in the nucleus accumbens and more instead going into the medial prefrontal cortex. So in other words, connectivity between the VTA and the nucleus accumbens is decreased in DCC haploinsufficiency. The data we have on this is 27 mice, 16 wild type, 11 mutant, in which they don't have a copy of the DCC gene. All the mice were male and all were on the same C57 Black6 background. The mice were actually imaged here, so we obtained it through a collaboration. They were imaged at 7 Tesla using a true FISP sequence, and the images were acquired at, I think, about 140 microns isotropic resolution. So shown here are images of normal control mice and an image of a DCC haploinsufficient mice. It's really hard to see any differences, but through image registration, we can pick up very subtle differences in volume. We wanted to look at structural covariance between the VTA and parts of the brain where neurons are sort of project to from the VTA, in particular the nucleus accumbens. And so we again went to the Allen Institute's viral tracing data. We picked out about 16 experiments in which they injected in the ventral tegmental area. We found all of these experiments, they projected to the nucleus accumbens. And then we essentially used image processing methods to binarize out these two regions and create a nucleus accumbens mask that's shown in the green and a VTA mask that's in the red. And we transformed that onto the study template. And from this, the idea is that we use, again, the Jacobian determinants that we get back from the image registration and compute volumes of these two different regions. When you look at the volumes, in particular, if you look at the volume of the target region, that's the VTA, sorry, that's the nucleus accumbens, you find that the volume is decreased as expected. That's because you have fewer neurons going there synapsing in the nucleus accumbens in the DCC haploinsufficient mindset. You also do see a slight decrease in the seed region. The VTA is a bit smaller. So I'm not sure, potentially that just might be there are fewer neurons in general, but the effect is much larger for the target. When you look at structural covariance between this pair of regions, though, you find that there's effectively no difference. Again, that goes against the hypothesis of wiring being required for covariance. So in summary, structural covariance in this genetic model of miswiring, it doesn't, in this model, you don't see a change in structural covariance. So this to us, again, the sample size is small, but collectively these experiments all converge to the same idea that wiring isn't necessary for the formation of structural covariance. So a lot of people were involved in this. I'd like to thank them all and happy to take any questions. There's one more?
One in the chat.
Oh, one in the chat.
I can read it.
Yeah, good job. Yeah, if you can read it out.
So if the data came from different experiments,
there could be a slight difference in age that can impact brain region involvement. Is that true? And if so, how do you?
So I'll answer that by saying that in the first experiment, we used all adult mice. When you haven't talked about, in which we've looked at structural covariance between specific regions, specifically the thalamus and the cortex, in which we've used mice that were almost entirely of the same age. They were all, again, controlled for strain. They were acquired using the same sequence. They were basically as identical as possible, and the results were kind of the same. You get these robust structural covariance networks, despite there being no variation in age. So the question is, where do you think you should go to figure out what's happening? There's many ways I can see this going. So one way to test this is to look at the role of firing. So I talked about wiring together, firing together, growing together. But you may not have wiring, you can still have firing through different pathways. So one approach would be to use functional data to see whether in the BTBR mice, does function change, and does that change map to structural covariance? The other thing you could do is you could use other models of disconnectivity, like the cuprizone model, in which you basically provide cuprizone, for example, to the diet to the mice, and that induces demyelination. And this would allow us to generalize to other tracks more than just the corpus callosum. That's a good idea. So the question was, have you considered sensory deprivation? We haven't, but that's certainly something we could try. Or the alternate would be stimulation of some sort.
The deprivation will answer a different question, like wire together, after together.
Potentially, yeah.
And so maybe, because I mean, one possibility is that your growth is that limited, right? And so it's an adequate mechanism of the best.
Yeah. Yeah, on my to-do list. So the question is, have you looked at cortex versus subcortex separately? In the experiments I talked about, or in the studies I talked about, we haven't. In studies on development and coordinated neurodevelopment that we've done that I haven't described, we've looked at trajectories of structural covariance separately within the cortex and the subcortex. And the answer to that typically is what you find is so across the brain, you find high structural covariance early on in life, and it tends to decrease over time. This is seen predominantly in both, well, it's seen both in the cortex and subcortex, but in the cortex, you do see a slight increase over time. So maybe there is some effect of coordinated plasticity, but it's still dominated by this mode of generally decreasing structural covariance.
Does the contribution of gene expression change
over time? Yeah, that's something that in principle I would agree with, but to assess that you'd need the right kind of data. So we have the gene expression data over development from the Allen Institute. They've profiled gene expression in both prenatal, 4 time points in prenatal life and 3 time points in postnatal life, but there's no connectivity data at those times. Okay.